Okay, hello everyone and welcome to GCI Research Rounds. I'm going to go ahead and get started with just some normal housekeeping and then let people trickle in as they're joining over the lunch break. So welcome everyone. As many of you know, my name is Stephanie. I'm the research coordinator for the Gynecologic Cancer Initiative. Before we get started, I'd like to just take some time to acknowledge that I'm joining today from the traditional unceded territories of the Coast Salish peoples, and specifically those are the territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And as always, I encourage folks to just take some time to reflect a bit on the territories that you're joining in from today. If you are interested in learning more about it, you can always go to nativeland.ca and learn more about the specific territories which you're joining in from today. So I'm just going to jump into some brief housekeeping. So as usual, please make sure that you keep your microphones muted for the duration of the presentation. Please also feel free to pop any questions that you have for Dr. Nelson in the chat, and we'll have a short Q&A session closer to the end of the session. This meeting is being recorded and it will be made available on our website a few weeks after today. Okay, so without further ado, I'm very happy to introduce our speaker for today, Dr. Brad Nelson. Dr. Nelson is a professor of biochemistry and microbiology at the University of Victoria and a professor of medical genetics at UBC. He's also the scientific co-director of BC Cancer's Immunotherapy program, and his lab is pursuing the development of innovative immunotherapies for cancer. So specific areas of focus that the lab works on is understanding how tumor reactive T cells and B cells promote survival of cancer patients, using genomics and bioinformatics methods to decipher the barriers to effective immunotherapy, and using the power of genetic engineering to create more precise and potent immune cells for use in immunotherapy. So I'll pass it over to you now, Brad. Thank you so much for giving the talk today.
Great. Well, thanks very much, Stephanie. Thanks for the invitation, and thanks to the whole GCI group for putting together this series, and thanks to all of you for joining over your lunch hour. I'll just acknowledge that I'm on Coast Salish territory as well. In my case, it's the Lekwungen peoples of the greater Victoria area. And yeah, I'm going to be talking about two things. The first half of my talk is going to be really about deciphering the immune response to gynae cancers. And I'm mostly going to be focusing on high-grade serous ovarian cancer, which is what my lab mainly works on, but I'll be referencing other cancers. And then the second half will be about re-engineering the immune response. How can we take what we're learning about the successes and the failures of the natural immune response, take those lessons and apply them to engineering designs that might ultimately lead to curative therapies for these diseases. So this first image is just really to whet your appetite. This is a multicolor immunofluorescence image generated by Katie Milne and team in my lab of high-grade serous ovarian cancer. And the different colored cells are different flavors of immune cell, helper T cells, killer T cells, B cells, plasma cells, macrophages, which I'll be talking about today. And the tumor cells themselves, the bad guys, are just the sort of this fainter coloring in here. So just to really kick things off by making the point visually that the immune response is not some esoteric concept. Immune cells, when they decide to attack a tumor, it's pretty obvious. You get very dense, brisk infiltrates such as these, and one has the impression, although this is a static image, but that if we played the take forward on this, that this particular tumor site is probably destined to disappear. Interestingly, we can see just right adjacent to it, another important point is we've got another tumor site, within a millimeter or so of this one, where we can see the immune system is struggling a bit and having less infiltration of cells into that tissue. So right away, also making an important point about the heterogeneity of these responses, even within a given patient. So I'll start the story by talking about T cells. T cells are wonderful cells. We all, I think, know and love them to some extent. And those who study cancer immunotherapy certainly know and love T cells very well. And that's because T cells have this incredible ability to be able to recognize and destroy cancer cells, and of course, other types of disease cells, such as virally infected cells. And this picture it's a scanning EM taken of T cells and cancer cells in a dish. But I like it because it shows how T cells work. They actually need to reach out and touch their target cells, make physical contact with them. And this is what T cells, how they spend their day, is crawling around the body, feeling other cells, and basically asking the question of each cell, are you healthy or not? And the way T cells are feeling is through their T cell receptors, their antigen receptors, and they are looking for, much like a blind person reading Braille, they're reading little physical bumps on the surface of the cancer cell. These are peptide epitopes presented by the major histocompatibility complex. If it's a healthy cell, the Braille code that's being displayed would be all words from the normal lexicon. Let's just use English as common language for this example. So they'd all be English words that are familiar to the immune system. If it's a virus-infected cell, you'd start to see foreign words appearing on the surface coming from the viral genome. In the case of cancer cells, you see spelling mistakes appearing on the surface. So typos in the form of point mutations or gene fusions creating novel junctional sequences, that sort of thing. And so T cells have to make the decision with each cell they encounter whether to let it live or die. There's three things that need to happen then for a T cell to make a decision to kill a cancer cell. First of all, and there's the three A's, which I hopefully makes it easy to remember: antigens, access, and activity. So there needs to be antigens expressed by the cancer cell that allow the immune system to distinguish healthy from tumor. Again, those could be the spelling mistakes arising from a mutation. There needs to be access. The T cells have to be able to get into the tumor because they need to be able to make this physical one-on-one contact with their target cells. And they need to have activity. So once in the tumor, the T cells, because they're living, breathing things, they need to be able to stay active. And so if there are metabolic issues that are slowing them down, or if there are immune suppressive factors in the environment that are suppressing them, then those things can also, of course, interrupt the anti-tumor response. But if you get these three requirements all lined up, all positive, what can happen next is the T cell will grab on very strongly to the cancer cell through adhesive interactions, poke holes in the cell membrane through perforins, and inject toxic enzymes such as granzymes into the tumor cell and cause it to dissolve within a few minutes of contact. So T cells, when they get serious about killing a cell, can do so very quickly and very efficiently. This is just another image, a multicolor IHC image, much like the one I started with, but just illustrating these points. So here, the good guys, the killer T cells that I've been talking about, are shown stained brown here. The bad guys, the tumor cells, are purplish. Then some other good guys are our helper T cells and B cells that are in there as well. And I like this just because it really makes the point, I think, these T cells just really do crawl into tumor tissue. They squeeze between the tumor cells, and we imagine what they're doing is reaching out and touching and reading those tumor cells, looking for antigens that would be a signal to kill. But importantly, when we look at a number of tumors from a strong immune responses, I also made that point even within a patient on my opening slide. What we see is, if you look at enough patients, is three major patterns emerge. So one is these hot tumors that I've been talking about. Immunologists call them hot because we get excited when we see lymphocytes like this. That's a brisk immune response, involves T cells and B cells. And to be categorized as hot, importantly, these lymphocytes need to be not just in the tumor stroma, but actually penetrating into the malignant epithelium, where as I've explained, they need to be in order to do their job. So that's the definition of hot, is that you actually have T cells in the epithelium. On the other end of the spectrum are cold tumors, which just lack lymphocytes in either stroma or epithelium, and they really do look this cold. Then there's the intermediate group, but which is, we think, a distinct class, which are the warm tumors, where you have the T cells and perhaps other lymphocytes in the stromal region of the tumor, but not invading the malignant epithelium. So these three patterns are seen not just in ovarian cancer, but really across all solid tumors. So it seems to be reflecting some fundamental interaction between the immune system and cancer. These patterns matter prognostically. So if we look at prognosis of patients and stratify patients based on these three patterns, we get the following results. And this is really a compilation of a number of studies that we published over the years from various folks in my lab acknowledged there. And this is for those among Oathcare, this is based on the OOU TMA that you've really generously provided to my lab for these studies over the years. The point here being that the really hot tumors that have all of the players present and in the epithelium have a much better prognosis, not good enough, but better than those with just the weaker responses, just T cells and those warmer tumors or the completely cold tumors shown on this black line. So when you get all of the players on the scene, the patient has a higher likelihood of survival. So this leads to questions, a lot of questions, of course, but one that I'll address is what's underlying the cold tumor phenotype? What's going on at those sites? And when thinking about this as immunologists, we think about what signals are the T cells receiving. I talked about antigens, access, and activity. So if there's, let's just assume there's antigens there, what might be affecting the T cells' activity? And so these are, this is a nice figure showing all the different receptors, or many of them, that mediate the dialogue between T cells and their target cells. So I've talked about the antigen receptor, the reading Braille part is shown here, but there are all these other receptors, some of which have a stimulatory effect on the T cell, if they're ligated. Others shown in red have an inhibitory effect on the T cell, if those are ligated. And so it's really a very complex dashboard of interactions between the T cell and the target cell, and all of these having some influence. Ultimately needs to be a binary decision by the T cell to either kill this cell or not. So all of this information needs to be integrated. So how do we make progress? Well, a recent study from my lab, led by Alex Rodriguez and Finn Hamilton, postdocs in the lab, who really came up with some really interesting ideas around this. This is just one figure from their paper where they looked across cold tumors in a wide variety of human cancers using the Cancer Genome Atlas. And they looked for immunologically relevant genes that were associated with the cold tumor phenotype. So these that in red would be genes that are upregulated on cold tumors. And there's a number of interesting leads here, but the one that we followed so far is second on the list here is the poliovirus receptor, or molecule known as CD155, which again seems to be associated with cold tumors. So back to this map, it's this pathway over here. CD155 on the target cell can bind to a molecule called TIGIT on the T cell, and that sends a red inhibitory signal to the T cell. So Katie Milne and her team came up with a multiplex IF panel that allowed us to look at this new molecule, new to us, CD155, in the context of other immune cells, and in the context of the, I think, quite famous now PD1 PD-1 pathway, the major checkpoint pathway in the clinic these days for immunotherapy. And what really jumped out at us is that CD155 in orange here is expressed when it's present, is expressed on the tumor cells themselves at and can be at very high levels and quite uniformly throughout the tumor bed. In contrast, PD-1, the more famous checkpoint at this point in time, and we had previously shown this, is mainly expressed by macrophages in the stromal region of the tumor. And that's this turquoise and purple pattern that we see through here. It's actually much less common to see PD-L1 expressed by the tumor cells themselves. So if you're a T cell trying to kill this tumor, it's quite a gauntlet that you've got to run through here. You've got to get past PD-L1 in the stroma, only to be hit by CD155 on the tumor cells. So two stop signs along the way that may need to be addressed. Interestingly, when we looked at a number of tumors and summarized the data, what we found is, and we had previously shown that PD-L1 is mainly expressed in hot tumors, and so that's shown here, whereas CD155, this new checkpoint, is predominantly expressed in cold tumors. Not exclusively, we find it in the other types as well, but definitely up on many of the cold tumors. And so one can see there's a complementarity to these two patterns. And we think this might be one explanation for why targeting just the PD1, PD-L1 pathway alone has not been very successful in ovarian cancer. This could represent one additional roadblock that needs to be addressed. And so, of course, then this provides rationale for dual targeting of these two pathways, the PD1 and TIGIT pathways, TIGIT being the ligand for 155, or the receptor. And so we've actually put forward a clinical trial proposal through the IPROC mechanism to the CCTG and the Cancer Research Institute. And we're hoping to have an arm of the IPROC study that will study this combination. So that's just a vignette about the cold phenotype. There's a lot more going on there for sure, but wanting to just give you a flavor for what we know and how we're approaching these problems. What can I say about what's going on with the hot tumor phenotype? Well, in a study that we did together with Alan Zhang, a graduate student in Saurabh Shah's lab, in collaboration with Rob Holt and with many people in OVCARE and the GCI as well, were involved in this. We did really a really extensive genomic and immunologic profiling study of tumor sites getting different spatial locations from within the same patient. So really looking at how the immune response varies at different locations, different tumor sites within a given patient. And there's a lot of information in this paper, but the one key finding that I'm just going to highlight today is that, and this is showing the results for a summary for just one patient in the study, where we had multiple tumor sites, which are illustrated by these cartoons here. And Saurabh's group was able to decipher the clonal phylogeny of the tumor cells within these different sites. And then my lab looked at the immune profiles that mapped to those different sites. And what we found, not just in this patient, but across patients, was that the hot tumor sites tended to be those that have more of a monoclonal or oligoclonal composition. So fewer tumor clones, like this one here, is mainly made up of clone B from this phylogeny. In contrast, the cold tumor sites tended to have a more polyclonal composition, as illustrated here, this site being made up of a number of different tumor clones from the clonal phylogeny. Now, this is just an association. We don't know cause and effect here by any means, but it certainly is suggestive of one hypothesis, which is this idea that Alan coined of immunologic pruning. And the hypothesis goes something like this, that perhaps all tumor sites start with some degree of polyclonality, though it gets ignited there and an immune response starts, then kill off most of the tumor clones. That's the pruning part, and just leave behind the one or two clones. Leads to certain testable hypotheses that we're working on now. But all is not well at these hot tumor sites, far from it. The hotter a tumor site is, the more likely it is that we're going to see evidence of antigen loss of the major histocompatibility complex, which is so key to that Braille reading mechanism that I talked about. So here, basically, you're losing your braille between these two things. We've also shown in other studies that the tumor-reactive T can die from patients at their first and second relapse. We've shown evidence that the tumor-reactive T cells actually can disappear by the time of second relapse in the study that I'm citing. The hotter the tumor is as well, the more immune suppressive factors there are there in general. I talked about CD155 being up on cold tumors. That's true, but there's certainly a lot of suppressive pathways like PD1 that are active in the hotter tumors. And also, we now appreciate that these hot tumors, I'm showing you all these images and lots of T cells in there, but it appears that the majority of those T cells don't even recognize the tumor. A lot of them are viral reactive T cells. They recognize viruses like CMV, influenza, and we think they're just drawn there because there's inflammation. They're just coming in based on basic inflammatory cues and not recognizing the tumor per se. And it turns out that it's probably a minority, like even less than 10% of the TIL that actually recognize the tumor. So the rest of them we call bystanders. They could be physiologically important. We don't know, but it doesn't seem like they would be given that they don't recognize the tumor. So this really, gives renewed impetus for this question of then what is the phenotype of tumor-reactive TIL and exactly how do these T cells work given all of these problems? So I'll just show a few slides on a paper that we just published in Clinical Cancer Research, and this was a collaboration with Christian Steidel's group and a number of individuals involved from my lab, Celine Lamont, Marcia Wooters, and Julian Swazinski, and others. But these are the three first authors. And so for a variety of reasons I don't have time to get into, we had some clues from the literature and from our own work that these markers, these three markers, might help define the tumor-reactive CD8 positive T cells. So help us tell the difference between the bystanders and the bona fide TIL. So the markers of CD103. And so our hypothesis going into this study was that T cells that co-express these three markers are the tumor-reactive T cells. That's the hypothesis. So again, Katie Mill and her team put together a multiplex IF panel to allow us to stain for these three markers, including as well as CD8. And then we looked at a large number of tissues. And when you do this, you get sort of this interesting jelly bean sort of pattern shown here, all sorts of different colors of jelly beans. And these are just T cells expressing different combinations of these markers. And the jelly bean of interest in this case are the yellow ones, which co-express, they're CD8 positive and they co-express these other three markers. So we think that we're hypothesizing these are the important ones that are doing the work. And when we did this then on the OOU TMA, again, the one I've showed previously, we found that really strong evidence that yes, these triple positive CD8 TIL have the strongest prognostic significance. So they not shown here, but they constitute only about 10% of the TIL overall. So they're a minor population, and yet they seem to be doing all the work prognostically. So in this Kaplan-Meier curve, I'm only showing you hot tumors. So all of the cases shown here have CD8 positive TIL, but now we're substratifying on these other three markers. And the purple line shows the TIL that have this triple positive phenotype. And you can see that when those TIL are present, those patients are doing better than the rest. So now we've got a handle on these tumor-reactive TIL, and we can ask more about them. And so this is where our collaboration with Christian Steidel's group became really key, because they helped us start doing single-cell RNA-seq on TIL from patients. And we are then able to zero in on or zoom in on the triple positive cells and ask, what's different about them? What genes do they express that's different from the triple negative cells from the same patients? And this volcano plot shows all the differences that we found. And there's obviously lots of them. And I won't go through them all, but you can see a blow up here of some of the key genes that are enriched on the triple positive cells. The summary statement on all this, what we learned from this is that the triple positives have a highly activated phenotype, which fits with the idea they're in there recognizing the tumor. It's a complex phenotype. It's got some surprises. There were about, so how many different T cell clones recognize the tumor? It's on average of only about 25 per patient. So that really puts an upper limit, if this number is accurate, it puts an upper limit on the number of antigens that might be present in these tumors. Not a whole lot. These T cells express granzyme B, which is involved in tumor killing. They express CXCL13, which interestingly is involved in recruiting B cells to the tumor. And they have a highly inhibitory type phenotype. They express not only PD1 and TIGIT, which I talked about earlier, but other immune suppressive markers, CD39, LAG3, TIM3, and so on. So it's complex. They're highly activated, but they're also showing lots of signs that they're being inhibited. So I kind of think of them as a car where you've got the foot on the gas and the brake at the same time, and you're kind of stalled out. Might be one way to visualize that. The B cell connection was really interesting. We've been studying B cells in parallel for a number of years, so it certainly caught our attention to see that these the bona fide T cells, one thing they seem to be doing is calling out for help to the B cell compartment. And that fits with the image that I showed you at the very beginning, where we've got these T cells in here in white, when we get these really hot tumors, we can see in green these B cells, which are also part of that infiltrate, we think being called in by the T cells for extra help. And then in the stroma of really hot tumors, we see a lot of plasma cells in red. Plasma cells are mature B cells that are the cells that make the most antibody. So they're the antibody producing version of T cells. So what are B cells all about? What role do they play in anti-tumor immunity? I think we're all acutely aware of our B cells these days with COVID and COVID vaccines. It's the B cells that make the antibodies that everybody's talking about as a measure of the efficiency or efficacy of vaccination. So that just shows that here you start with a B cell, and if it's properly activated, turns into a plasma cell, which makes antibodies. So here we've got this going on in the tumor microenvironment. And so these plasma cells are present. We have evidence they are making antibodies. We don't know yet what the antibodies are against. That's a project in the lab that Alison Vanville is leading. But this is from a review that Celine and I wrote recently, shows some of the things that antibodies can do to hurt a tumor. So they can block the function of the antigen they recognize. They have a way of actually enhancing T cell activity by increasing antigen presentation through a process known as opsonization. They can also trigger complement reactions, which can have a direct killing effect against the tumor. They can engage natural killer cells through a process known as antibody-dependent cellular cytotoxicity. So you get your NK cells attacking the tumor, and they can get the macrophages attacking the tumor through a process known as antibody-dependent cellular phagocytosis. Real mouthful there. But one gets the impression then that if you have antibodies on the scene, all sorts of orthogonal effector mechanisms can start to kick in. And that's what we think is happening in these tumors, and these are the hypotheses that we're pursuing. So that's the deciphering part of my talk. And so in the last half, I'm going to talk about, okay, how can we take these lessons? And I've just given you a few vignettes, but how can we take what we've learned and try and do better than Mother Nature and actually re-engineer the immune response to the patient's benefit? And engineering, most of the work these days is focused on T cells, although I'll just, before I forget to say, there's also engineering efforts with NK cells, macrophages, B cells. There's lots of exciting ideas out there, but I'll focus on what's being done with T cell engineering, because that's where the most activity is. So there's three major ways of what we call T cell products. types of T cell products you could imagine giving patients to try and treat their tumor. And one is a natural TIL. So just taking these tumor-infiltrating lymphocytes that I've been talking about, take a surgical specimen, you can actually extract those T cells, grow them up to large numbers, and give them back to the patient with various immune modulators to try and just really amplify the TIL response. And I'll show you a bit of data on that. A second is to genetically engineer the T cells to express a T cell receptor that recognizes the tumor. So this is taking that Braille code and really hardwiring it into the T cells so that for sure they ought to recognize the tumor. And then CAR-T cells are a strategy with much the same goal of engineering the T cells so that it is hardwired to recognize the tumor. But here it's done through an antibody-mediated construct rather than a natural T cell receptor. So TIL therapy, and I'm going to now just really focus on gynae cancers. TIL therapy was developed in the melanoma setting, metastatic melanoma, and has proven to be very successful and is on the way to having the first commercial product made by a company by Iovance to treat melanoma. So there's been really quite considerable success there. In the gynae cancer space, not so much yet, with the exception of work in cervical cancer. And this is a real key paper that I would point you to learn more about this. But in cervical cancer, and this is work that's been done at the US NCI primarily, seeing on the order of 28% objective response rate in metastatic HPV associated cervical cancer. And in this study, 2 of those responses were complete and ongoing, quite durable. So in the T cell therapy field against solid tumors, this is actually very exciting because it's a step now from the melanoma setting, which might be a special case, into the more common epithelial cancers. And to see these kind of signals is encouraging that the field might be on the right track. But there's a lot of logistics around TIL therapy, like needing to get a fresh tumor sample from patients and stuff that make it, difficult to implement in the clinic. So that gives reason to explore engineered T cell receptor therapies, where you can take just a patient's blood sample as a starting point. You don't need the tumor, just the blood sample, and just engineer it to have the receptor that recognizes the tumor. And here too, the same group at the US NCI, Christian Heinrich's group, has really been a leader in this area. So they've got a T cell receptor that recognizes the E7 protein from HPV and conducted a trial then of giving those T cells to try and treat these cancers. And this is a summary of the data from the trial. So these are all metastatic tumors. Interestingly, 8 of the 12 patients had received prior anti-PD1 therapy. So they had been failed by checkpoints or were at a point of failure with conventional checkpoint therapy. There was cyclophosphamide preconditioning, sort of standard dose of engineered T cells given, some amount of interleukin-2 as a growth supplement to the T cells, and quite an impressive response rate. 6 out of 12 patients experienced a partial response, and 4 out of 12 having stable disease. Again, a real positive sign that we're finally making progress against the solid tumor microenvironment. And in the same study, they looked at some of the resistance mechanisms, and those were related to some of the limitations of T cells I talked about earlier. So loss of antigens, MHC, and interferon response pathways. So I'll just switch now over to CAR-T cells and say a bit more about those. So again, this is another engineering strategy. The basic idea, there's different generations of CARs, but what they all have in common a T cell, and that antibody can be directed to whatever your favorite antigen is, and that will hardwire the T cell to recognize cells expressing that antigen. And then where the different generations come in is what intracellular domains are part of the CAR. What kind of signals do you want to send the T cells? So there's different varieties there. So CAR T cells, where they've had their biggest impact by far, is against CD19 positive leukemias and lymphomas. And there, in the ALL setting, they achieve reproducibly about a 90% complete response rate in both pediatric and adult cases. So really just astounding clinical results. In lymphoma, where you have a more solid tumor microenvironment, the response rate is not as high, but it's still substantial and very promising. This has led to FDA approval for a number of commercial CAR products, as well as Health Canada approval. Where we stand in BC at this moment in time is these are Health Canada approved products, but our province is not at this point reimbursing those. And so access to these novel therapies is an issue, which I'll talk about in a minute. So starting from the success of CD19 CARs, though, what would we like to see next as a field? We'd like lower toxicity. There are toxicities associated with this therapy that we'd like to address. We'd like to apply this to other types of cancer beyond leukemias and lymphomas. We'd like better penetration of solid tumors, not only the lymphomas, but if we're going to get serious about epithelial cancers, this is a big barrier to overcome. We need fine-tuned control mechanisms. Today's CARs, once you put them back into the patient, we have very little control over their activity. Some control, but not near as much as we'd like. So we'd like to be able to speed the CARs up or slow them down as needed. We'd like to be able to stop them too. If the patient's tumor is gone, we'd like to just stop the reaction, or if there are serious toxicities, we'd like to be able to stop it as well. So we need mechanisms to do that. And all of this needs to remain affordable, feasible, and sustainable if patients are ever to benefit from these therapies. So I actually think we can achieve all of these goals, and it's a very exciting time to be working in this field on all of these fronts. I'll start with the just tell you what we're doing in Canada and BC. In particular, we've put together a consortium we call CLIC, Canadian-led Immunotherapies for Cancer, and it's a collaboration between a number of us at BC Cancer, the leads being Rob Holt, Kevin Hay, and myself, and then a group in Ottawa, Natasha Kekri, Harry Atkins, and John Bell being the PIs, where we've put together a CAR-T program that allows us to manufacture CAR-T cells from scratch, basically starting with a whiteboard design all the way through to a clinical product, all done within the academic sector. That was our goal. And we started by just making a rather generic version of a CD19 CAR-T cell. So nothing clever scientifically, but that just allowed us, by starting with a product that we knew ought to work, it allowed us to really focus on all the implementation pieces, which were substantial. It took a number of years to put the pipeline together and open our trial. And one of the key things we're doing here is really thinking about access and affordability. And we think the future is in point of care manufacturing. We see a world in the future where every bone marrow transplant unit in the country would have a CAR-T cell manufacturing equipment in it. And this would just become, just like we do stem cell transplants now, CAR-T cells, no big deal. That's, we think, how access and affordability can be addressed. So with that in mind, we built a clean room at our research center in Victoria with funding from BC Cancer Foundation, CFI, BC KVF, a number of sources. And this is showing the team. This is showing some shots from in the clean room. And the instrument we're using is shown here. It's made by a German company, Miltenyi. It's called the Prodigy. And it's like a bread maker that sits on the counter. And as you can see, this is one of them here. You hang a bunch of things in IV bags, patient blood sample, some media, some beads to activate the cells, your viral vector that carries the CAR. And then you hook up this tubing set for each patient. And there's a number of pinch valves here. There's a magnetic separator. There's a centrifuge chamber, et cetera. You get the thing wired up, push the start button, and it goes through a 12-day fully automated closed manufacturing system to give you a CAR-T cell product out the other end 12 days later. And so by doing it this way, you can have several instruments in our facility working in parallel, and then a very dedicated team of people. It's not quite as simple as just pushing start. There's a lot of work that goes into setting this up and then monitoring things and then checking the product in the end. It's actually a team of about 20 people involved in doing this. But the net result is really quite elegant, that you can start with blood at the beginning from the patient, and 12 days later, you've got an IV bag of CAR-T cells ready to infuse. So we opened our trial in 2019. So we're 2 years into it now, phase 1/2 trial, so safety and efficacy. This is in collaboration with the Ottawa Health Research Institute. They're the actual official sponsor of the trial, and the manufacturing site is our lab in Victoria, BC Cancer. Patients are being treated at Vancouver General and the Ottawa Hospital, and we've now completed our phase 1. So we've treated, we're actually past our first 20 patients now, and we're into the phase 2 portion of this. And the data is still being analyzed and embargoed, that sort of thing. But the high level results I can tell you is that we're seeing the expected rates of efficacy that we expected going into this. So we're very encouraged by the results we're seeing so far, and we feel that that our methods our manufacturing methods are working well. So in just the last few minutes, what can I'll just tell you a little bit about how researchers are trying to tackle some of these other biological challenges of CAR-T cell therapy. And so this is a very, very active area of research. This is a nice review article for those who want to read more about it. What this figure is showing is that here's a CAR lying on its side now, just to illustrate that on where the antibody normally sits, there's all sorts of interesting designs, ideas being tested in place of just single chain antibodies, including nanobodies, cytokines, et cetera. So the recognition piece, there's lots of cool ideas being worked on there. The signaling side of it as well, there's lots of different signaling domains that are being tested. So this really gets down to what instructions does the CAR send to the T cell. And then in this area, the idea here is that if you're engineering a T cell anyway, you're going to put a CAR in. Why not put other things in with it that might help the T cell? So for example, you can co-express enzymes that will actually chew into the extracellular matrix, so that the CAR T cell, if it's having trouble penetrating the tumor, can it chew its way in by secreting to mimic the effects of these cells, trigger all the good things that come from an antibody response. So lots of very innovative ideas being tested, and now literally hundreds of companies around the world and academic groups working in this space. Another neat thing with CARs is that you're not limited to just one CAR. So you can actually put as many as you can fit in a lentivirus. So really up to 3 would be quite easy with today's methods. And you can wire the CARs depending on their signaling domains to create different logic gates. So you can have, you can put CARs in, CAR A and CAR B, let's say, and wire it in a way that the T cell will fire if either CAR A or CAR B gets activated. Or you could have it be an AND function, so that the T cell only fires if CAR A and CAR B are activated. You can have NOT functions, so the T cell would be activated if CAR A is ligated, as long as CAR B is not activated. So there's not many drugs that I can think of that allow you these kind of Boolean logic gatings. And I think that really opens up the possibilities for how we can wire T cells to recognize complex targets like ovarian cancer, where we don't have a perfect marker like CD19 on the surface. By Mark Wooters, and the project was completed by Alison Banville in my lab. looking in high-grade serous ovarian cancer, kind of a survey project really, of looking at 3 antigens of interest for CARs, CO125, mesothelin, and folate receptor, and looking at their expression using multiplex IF, so that for each individual tumor cell, we could score it as for expression of each of these 3 markers. Starting to think about what kind of Boolean logic CAR would make sense in this disease. What do we want, an AND gate? Do we want an OR gate? That sort of thing. And the net finding from the study was, of these 3 antigens, the best strategy that we saw would be to have an OR gated CAR that targets both CA125 and mesothelin. We thought that was about the right balance of good recognition with less toxicity. I've also been involved and had my first startup out of BC Cancer, working with a scientist, Marty Boulanger at UVic. We co-founded a company called Immunotherapies Therapeutics. So I declare my conflict of interest. I am a co-founder of the company. We spun this company off a couple of years ago. And what Immunotherapies is doing is not working on the recognition side, but more how do we control the behavior of T cells once they're back in the body. And what we've done is come up with an engineered cytokine receptor system, where the upshot of it is that after we give a patient CAR T cells, we can give them our engineered cytokine, and it will only stimulate the CAR T cells expressing our engineered receptor. It won't stimulate any other cells in the body. So it gives us a way of talking directly and exclusively to our CAR T cells or any T cells of interest. So we've got a direct line of communication. And then we've got a whole toolkit of intracellular domains on these receptors that allow us to send different types of messages. And so depending on the context, we might want to tell the T cell to just proliferate more. In other settings, the message might be kill, time to kill. So the idea is when a T cell hits a tumor, and if it's just left on its own devices, it might see too many stop signs. There's TIGIT there, or CD155, I don't want to go forward. We can send a message, no, push through, keep going, and kill. So that's the goal. And we see applications for not only CAR T cell therapy, but a number of other oncology and infectious disease and autoimmunity, regenerative medicine type applications. And this is just a bit of data from a study that we did over the last year. The work is being done at BC Cancer under a collaborative research agreement. And this is a mouse model for breast cancer, where we've got a T cell therapy that works pretty well. It gives partial responses in this model. And we then ask the question, if we install our receptor in these T cells and give them our engineered cytokine, can we do better than a partial response? And we did. We got results we're very pleased with, where showing here in each line here is an individual mouse, but showing in 4 to 4 mice in this study, when we treated them with our engineered cytokine, we got really massive T cell expansion as measured in the blood. And that was associated then with tumor regression in 4 out of 4 cases. And these were durable, complete responses lasting out to 80 days when we ended the experiment, whereas the mice just treated with vehicle had the partial response we were expecting, but then relapse. So just to wrap it up, where are we at today with engineered cell therapies for gynae cancers? Well, as I briefly reviewed, TIL and TCR therapy are showing modest success in cervical cancer. I think a really exciting lead there. In fact, this is ahead of other solid tumor sites. Cervix is actually one of the showcase examples of cell therapy starting to work. CAR T cells have been unsuccessful so far, largely, except for the occasional anecdotal report. And the challenges there are the paucity of tumor-specific antigens, although I did talk about how with logic gating and stuff, we could maybe address that. And then in general, the tumors have ways of blocking T cells from entering, or they shut them down once they're inside. I talked about that gauntlet of PD1, CD105, et cetera, that T cells face. And tumors are continually evolving. And the more selective pressure we put on them, we can expect them to try and evolve their way out of it. So that's a challenge, not just for immunotherapy, but for all therapies. what are things going to look like in the coming decades? Better guidance mechanisms to help T cells recognize tumors. I've talked about some strategies there. Throttles and gears to allow more potent signaling by the T cells without exhausting them or causing safety issues. I really believe T cell therapy, these aren't just lone wolf hunters that we're sending in. They need to work with the immune system, the patient's immune system, to get an orthogonal or multi-pronged attack going. I think braking mechanisms, there's already examples of good brakes coming out. Automated manufacturing, I think, is the way to deal with cost and accessibility issues. And so I really do think it's achievable for us to come up with effective, safe, and affordable cell therapies that leverage the host immune system. And I think here in BC, we've got a lot of things going in our favor for making a real impact in this area. There's my work, but what I'm really referring to here is what's possible within the context of BC Cancer, UBC, and the GCI initiative, bringing all that expertise together. And I'll stop there. And I've recognized people all along, but obviously there's a lot have been involved in this. And I guess I'd just like to, in addition to all the collaborators, I'd like to thank the funding agencies, and most of all, thank our patients and care providers who've provided access to the samples and data and inspiration for our work.